Hello everyone and welcome to the second semester of Anatomy and Physiology here. We're going to begin with the first unit here, which is Chapter 18, which is the Intro to the Endocrine System. And so Chapter 18 is going to cover the entire endocrine system. And as you can see here, I broke it up into two parts. So this first lecture here, part one, it's going to be a little bit longer because I'm going to be breaking down how my lectures are set up, since this is the start of essentially the second semester if you're taking an Anatomy and Physiology course. And as you can see, there's going to be two groups of individuals that I talk to. I'm going to be talking to the students, my students specifically, on the important things that I expect you to know. And then occasionally I'll tell if there's anyone watching, any other guests watching, I'll tell them sort of and give them hints as to some importance or some things that they might want to check if they're taking a course with another instructor. So we might have two audiences here. It won't interrupt the flow too much, but just understand this first lecture here is going to take a little bit because I'm going to be explaining things and how my lectures are set up as well. But on average, most lectures or most parts of a lecture are going to take about 30 minutes. And here you can see this is broken up into two parts. So you can expect lecture one being 2 parts long. It's going to take about an hour, 30 minutes and 30 minutes. That's kind of what I shoot for. All right. Why I broke it up into 2 parts and saying it's one lecture. One lecture is enough content to really cover concepts that I feel like students can sort of work through in a good couple hours of studying. And so I broke each into lectures, but I didn't want to do an hour-long lecture total, so I broke it down into 2 parts. And you're going to see this throughout. So moving forward, you'll see when we move on to lecture 2, I believe lecture 2 is broken down into either 2 parts or potentially 3 parts in some cases. But basically, each of this, when we complete both of these parts here, that's going to be all of what I refer to lecture 1. So lecture 1, part 1 is what we're going to be covering today. And so as you can see, lecture 1, part 1 would be the introduction to the endocrine system. Then we're going to get into the forms of cell-to-cell communication. It's not just the various forms of communication like we, of course, spent a lot of time. Those of you that have taken structured anatomy courses, most anatomy courses in the first semester, they usually end with the nervous system. So we know there's nervous system communication, but we're going to look into the different ways of chemical communications outside of that. So we're really going to define those. And then we're going to really just define 3 classes of hormones. So that's really the big picture that you're going to see in this first part. And like I said, it takes about 20 to 30 minutes on average to break this down. And then from there, you can see in part 2, we're going to overview all the endocrine organs, spend a little bit of time talking about their big picture functions. That way I can start, giving some quiz questions on what do these organs specifically do. And then eventually we're going to go into a deep look into the pancreas. And we're going to see that I follow along with Pearson's. In fact, I'll get to that in just a second. Okay. But I do not follow along with, I said I follow along with Pearson. Just understand that I'm going to hop around in the textbook. I'm going to cover topics in a way that I think makes the most sense for flow and having students understand it. So it might be a little bit different. I'm not going directly through chapter 18, and I do this occasionally. So different points I'll change. And you're going to see me actually put up the citations for which Pearson book we're following along with. All right, but in my lectures, you're going to always see that I begin with the objectives that you're going to cover. If you were to look at these objectives, it would be a great tool to make kind of like a note card. You can literally make a note card that's that first bullet point. And could you come up with a really good answer and spend some time on that? And if you just went through that list, I would say that's a really effective way of using the objectives that are in this list. That's a good way to see, do you know this information? All right. And then when we go down here, you'll see we get into the second part. Part 2 here is going to discuss what we're going to, of course, cover in the next posting, which is going to be lecture 1, part 2. All right, so let's get into this. The citations, here you can see chapter 18. I'm gonna make use of most images come from this textbook, which is written, the authors were Martini, Nath, and Bartholomew. And like I said, this is a Pearson book. It's the Fundamentals of Anatomy and Physiology. We're still following right along with the 11th edition, which of course follows most editions. Figure numbers might be slightly off. All right. So that right there is a typical introduction. I just want to give you a heads up. That's usually how I introduce some topics. But now, as you can see, the first slide here has the endocrine system. You can see it's a different color. It's bolded. It's underlined. So we're going to start describing it. So anything on this one slide, and it's just this slide that's red, is going to refer to the endocrine system. And again, it's a per slide, not per lecture basis. So the next slide, I use a color code scheme. And that color code is for that one single slide, unless I say otherwise, which is really, really rare that I would do that. I just usually stick to one slide, one color scheme per slide. All right, so let's think of the functions of the endocrine system. If we're going to break down functions, I always like these questions, and most instructors in most classes that you're going to take with anatomy and physiology, you should be able to describe the functions of any system we talk about. And so when we bring up the functions of the endocrine system, the main function is to regulate long-term processes, and it does so by maintaining chemical signals kind of in a range. You've talked about homeostasis before. You should know that homeostasis is, maintenance of our body in a range, right? Certainly 98.6 degrees is our kind of normal temperature, but we actually fluctuate throughout the day. We're going to be in a range, okay? So know the endocrine system regulates long-term processes. Just a good idea for long-term process to kind of make sense of when we talk about long-term processes. Think of the whole changes that occur during puberty, right? Puberty hitting, right around 12 to maybe going through 18, 19-ish. That whole range of puberty there is all regulated through this endocrine system. And that's what we mean regulating long-term processes. Or you can think of your blood pressure being 120/80 in the long term, like throughout the day, it's going to be an average of 120/80. That's the maintenance of a long-term process. All right. Our blood glucose levels, we try to maintain them to give us energy throughout the day. Okay, that's what the endocrine system does. And the chemical signals are very specific. Those, hopefully you were able to come up with an answer. What chemical signals does the endocrine system use? When we looked at the nervous system, right, the nervous system relied on neurotransmitters. If we said neurotransmitter, you should be thinking, okay, nervous system. Similarly, endocrine system has its own special chemicals known as hormones. And these hormones, if you notice, this is a new definition. It's in blue. It's a chemical signal. That's a hormone. It binds to, notice receptors are going to be a new definition here. It's going to bind to specific receptors between cells, and that's going to help, these receptors will relay information and instructors, as well as obviously the hormone. Without the hormone binding to the receptor, the messages it receives. So receptors are needed, but hormones are actually what are going to cause essentially a chemical reaction to occur in most situations, as we're going to see. All right. So endocrine system, long-term process by making use of hormones. What are hormones? Hormones are chemicals that bind to receptors that eventually lead to some sort of change. All right, so receptor, it's an effector. The effector is going to be, we go back to homeostasis. Homeostasis involves 1, a receptor, 2, a command center, and 3, an effector. The effector is what kind of carries out a response. And so receptors are the effectors. They're embedded in an effector's membrane protein that really responds to a signal. So a receptor is a membrane protein that usually responds to a signal. There's the chemical signal, or in this case, the hormone, will bind to it. This is how certain hormones target certain organs. It's because some organs have receptors for a hormone and other organs lack those receptors for that hormone, and therefore the hormone will bypass those that don't have receptors. All right, so examples of receptors to be aware of are hormones in general, light. We have photoreceptors. We have antigens. Antigens can trigger our receptors. I shouldn't say antigens. It's antibodies. Neurotransmitters can function as a chemical messenger. So we have receptors for neurotransmitters. We have receptors for ions. So in this case, these are things that can bind to receptors, is what these are examples of. So off to the side where it says example, things that bind to receptors. Hormones, light can bind to antigens is correct. Antigens bind to receptors. Those receptors would have been found, we're going to learn, in the immunoglobulins, which are, as we're going to see, antibodies. All right, and so the main mechanisms of intracellular communication are found below. So notice this has a lot going on a slide. I have color coordination. In this corner, this was the section of the chapter that we are looking at. This was all information from 18.1. You can see there's table 18-1 here, and you can see I have this figure. And so we're going to describe this information here. But so far, this is the easy content that you really need to understand. Hands down. Can you describe endocrine system? Notice receptors were concepts from prior material, so you really need to have a good grasp of them. And I like talking about them to remind you, hey, these are concepts you should have already known. So these are the main points of each type. So notice direct communication. You should know direct communication is a form of intracellular communication. And notice it's going to use gap junctions. So it's really easy for me to just ask, which of the following forms of communications strictly relies on gap junctions of neighboring cells? And a gap junction literally is your cells need to be sewn together in sheets, i.e. it's mostly going to be in epithelial tissues. And so notice, usually limited to adjacent cells of the same type. So make note that when it comes to direct communication, we're using gap junctions, and then really know the examples of epithelial tissues. And it's also found interestingly in the heart. In fact, the heart is to me, the most obvious form of this being direct communication, using those intercalated discs. And we're going to learn about this when we get to the cardiovascular system, but that'll be unit 2. So we're going to come back to this, but understand direct communication is using gap junctions to basically allow exchange. And certain ions like calcium, you should know calcium was a big ion that caused changes. Well, that's what we're going to see in the heart, right? Calcium was important for the release of the neurotransmitters, if you remember. It was important for causing muscular contraction. So that's when we think of direct communication. Those are the sort of concepts that we're thinking about. All right. Now, paracrine communication, when we talk about this, notice the mediators, the types of chemicals, are actually called paracrines. We only have a few examples of this, but basically what you need to understand that this is local area. So this is local communication. All right, notice paracrine concentrations are relatively high. Target cells must have appropriate receptors. So we're not going to use gap junctions where, since there's gap junctions, literally we didn't need receptors, but here we do. All right, and so understand with paracrine communication, it's communication that occurs within a tissue. And note, it's the healing processes. All right, so the healing process is one of the main examples of paracrine communication. You can also think not only healing, but same with fighting off infections and, of course, immunity. And we're going to be getting into that as well. So that tends to rely heavily on paracrine communication. So again, when it's paracrine, it's a simple definition. It's communication that occurs within tissues. And great examples are going to be the healing process. It's going to be local communication. Now notice we also get to autocrine communication. You got the prefix auto, and that should be a giveaway here, right? And so auto, notice, still using extracellular fluid, so it is the release. So we're not on gap junctions. We're just in the extracellular fluid because it's still going to be local, but in fact, auto means self, so it literally means target the same cell. And again, we're going to build off of this later, so we're going to build it off intercalated discs. If I tell you we're going to build on these concepts later, these are my signals to you that you need to know this, and it's likely going to be tested on. So autocrine, you should definitely know it's cell to cell, it's the same cell stimulation, and really the only time I know this is white blood cell stimulation, specifically helper T cell activation. So helper T cells, once they get activated, they release chemicals, and those chemicals activate themselves, and in some cases will also activate B cells, as we're going to see. So note helper T cells, specifically the word helper T cell, and sometimes I simplifies that as white blood cell stimulation. That's what WBC stands for, is white blood cell. So in either case, so understand from a teacher perspective here, you need to define autocrine communication as the type of communication that it's when a cell releases a chemical and stimulates itself. I'm giving you the example, or a specific example, helper T cell. But since the helper T cell is a white blood cell, this statement is also true. This is a more accurate statement, but on a quiz or an exam, if it was generic, I expect you to know it right now, generically or up to this extent. We will expand on it later because guess what? The second chapter of this unit is actually going to be the lymphatic system and the immune system. So we're going to be covering that later. So get familiar with this is all I'm saying. Now with endocrine communication and synaptic, these are the two that you really need to compare and contrast. So for these, if you kind of have my major bullet points on what I've described here, that minor detail, and kind of get that to memory, you're in good shape. With endocrine and synaptic communication, I fully expect students to have a really great understanding on this. And so notice, endocrine communication, it's going to be when chemicals get released into the bloodstream. If it's in the bloodstream, it is endocrine communication. And this is going to be really important when we get into talking about the hypothalamus, because the hypothalamus has neurons that are going to secrete, they're essentially neurotransmitters into the blood. But since it's going into the blood, it's actually not a neurotransmitter. It's going to be a hormone, even though it's going to be produced by a neuron. We'll get to that later. I know that might have sounded confusing, but if we release a chemical substance into the bloodstream, it is a hormone. It's that simple. Chemical signals in the blood are likely going to be hormones. I'm not going to say always are hormones because there's going to be exceptions to that, but often are going to be hormones if they're in the bloodstream. So when it comes to endocrine communication, you should know it's the targeting of distant tissues, because these were all nearby. This is literally the same cell, and this is literally within tissue. So endocrine is defined by targeting distant tissues, basically by using the bloodstream. All right, and then again, you should think long-term homeostasis or changes, i.e. maintenance of blood pressure, maintenance of blood glucose, maintenance of pH, maintenance of blood volume, maintenance of ion concentrateness. Notice endocrine system is maintenance throughout your entire life. And that's very different when we talk nervous system. Nervous system was the immediate effects, right? You open your eyes, you activate photoreceptors, and you start processing and creating vision. That's what the nervous system does. Instantaneous, right? Endocrine system, long-term maintenance. That's what you should think. Synaptic communication, of course, we know we associate with the nervous system. So you can think of it as neural communication or synaptic communication. This is within synapses, and it's the use of neurotransmitters. Neurotransmitters are the chemical signal for neurons. Hormones are the chemical signals that travel in blood. Paracrine is chemical signals within local tissue, which often you can put here where it says extracellular fluid. You can put extracellular fluid, or you might want to add the term interstitial fluid. I often use that. All right, but notice neural communication or synaptic communication is rapid. It's very fast, and the responses are very short-lived, like emergency responses, things like that. We're processing the changes. You want to move, you're moving from one location to another. That's all neural. So make sure you spend some time breaking this down. Long-term communication, traveling through the blood to maintain long-term processes. Fast, immediate changes. This is a great slide to study. It's worth understanding. It's really, this is like the simple questions to get right. This shouldn't be challenging content. I'm not saying you're going to necessarily grasp it immediately, but this should be something that, you look at 2, 3, 4 times and it really starts to click and you get the main points on it. Don't miss this. Spend some time really getting it down, because these are the simple questions you want to make sure you get correct. So this is going to take us now to figure 18.1, as you can see up here. And again, we're just doing an overview of the endocrine system, a general introduction here. And as you can see, these are all of the major organs you're going to need to be familiar with-ish. And I say ish because some of these hormones we briefly talk about here. All right, but continuing on with our comparison of the endocrine system versus nervous system, when we talk of the endocrine system, the strength of that signal is completely based off the concentrations of hormones within the blood, i.e., many of these hormones get maintained within a specific range, okay? And so we're going to be dealing with ranges, as I keep stressing, and that's what's going to be what maintains kind of blood volume, what's going to maintain blood glucose, those sort of things, all right? So when we need a bigger response, let's say we're in an emergency situation and we need a stronger response, well, the endocrine system would increase the concentration of those hormones. For instance, think of adrenaline, right? That's epinephrine, norepinephrine. And during an emergency situation, that signaling will be really high and maintained high to keep kind of that energy up. Okay, that's how to think of endocrine signaling. When it came to neural signaling and neural strength, that strength of signal was completely based on the frequency of neural input. So if you're lifting something light, there's minor signals and a small subset of signals being sent to a small subset of muscles. Whereas if you need to lift something heavier, you were going to increase the number of fibers as well as how often those fibers receive a signal. So that's kind of how the nervous system works. We recruit more cells as well as increase the frequency of signaling. So again, like I've said, comparing the endocrine system with the nervous system, especially because literally we're going to have, these are the two main systems of communication. We're spending an entire chapter on them. You should really be able to compare and contrast those. All right, but we'll be breaking all of these down later. I don't want to get too far into this. These are just a general idea of what you're going to see. Now, what I would encourage you to do is to revisit this figure. It's introduced to you really early on. It's the first figure of the book, and at that point, you're not going to really know much about any of these things. Certainly, you should be able to identify them. If you are taking an anatomy course or if you're in my actual class, I have plenty of these board models that basically it's That's a 3D version of this, and I can easily label up any of the things that they're pointing to, and say what is this organ, name a hormone released from that organ, or what's the general function of those hormones. But we'll get back to how I can label this up. But just keep in mind, check the lab when you come in for lab time to make sure you know what specific models we have, and be sure to verify what sort of test questions on the lab that I can ask using things like this. All right, but I'll clarify that along the way as we keep moving through lectures here. All right, so let's get a little bit deeper into hormones. And at this point, you can see it's red, so I'm defining hormones. And you're going to see the definition for hormone is going to change, and your definitions should change as well. When you make note cards, I would encourage you, if you make note cards, to continually update your note card with your understanding and key points that you want, instead of just sticking and going through the motions of reading a definition and trying to get the right term. All right, but when we go into hormones, understand what we're talking about: chemical signals that travel in the bloodstream that bind specific receptors of an effector in a distance. That is a really great overall definition. If it doesn't make sense to you, or if this seems like too complex of a definition, that's because I've used literally all of the key terms: bloodstream, receptor, effector, and distance, all in one definition. All right, but you need to get to the point that something like this makes a ton of sense and isn't too hard to understand here. Now, what is the function of the hormone? Well, functions of hormones are to modify cellular activity of the effector that they bind. So that's how hormones work. They bind to a receptor, and that's going to lead to some sort of change, often a chemical change that usually increases or in some cases decreases the activity of that effector. We're going to focus on the increasing the activity of the effector in most of the cases that we talk about here, okay? And the way it's going to increase cellular activity would be we can alterate the gene activity. In many cases, turning on a gene or turning it off. All right. In some cases, especially with things like adrenaline, we're going to see thyroid hormone. We're also going to see something called cortisol or cortisone. We have the stimulation of metabolic activity, i.e., the cell becomes more active and is basically using and processing energy is the way to think about it. Just think more cellular respiration is often how I think about metabolic activity. But it also involves the storing of, glucose to glycogen and kind of building up storage molecules as well. All right, and then changes in membrane permeability. This is important. This is what allows the entrance and the release of substances, which often leads to a cellular response. Again, if you're like, well, what's this mean, change membrane permeability? How does that lead to a cellular response? Like we've been seeing, and like I'm going to continue to stress, you should have finished the nervous system. If you've taken Anatomy 1, and you know that in order for the release of neurotransmitters, that required calcium to be uptaken by the axon terminal. That calcium uptake is a change in membrane permeability of calcium, and that calcium that allowed the entrance of calcium, and that led to the cellular response, which was exocytosis of the neurotransmitters. Okay, so these concepts you kind of have to make sense of. When ingredients come into the cell, just think it's causing a cellular response. All right, but the main point here is the overall function is to regulate long-term complex processes, which things like reproduction, things like digestion, chemical balance, our energy levels, all of that is maintained through the endocrine system and not the nervous system. The nervous system is using energy. It's really not processing energy the way when we think the endocrine system does. That's what revs up the cells. All right, and so the final kind of concept that we have to get through to close out the first part of the first part of lecture 1 here is the 3 classes of hormones. And so hopefully you can get familiar enough and to be able to list these pretty easy. But we're going to talk about 3 classes. We have amino acid derivatives. That's going to be your tyrosine derivative or a tryptophan derivative. Those are the 2 amino acids that can form something called amino acid derivative hormones. Tyrosine derivative or a tryptophan derivative hormone. And we'll get to those. All right, then we have something called peptide hormones. These are long chains of amino acids. You should know peptide is amino acids. That's all I expect you to know. Peptide hormones, chains of amino acids. You might want to put off to the side of my notes that this means, in most cases, notice the word acid on there. This is a polar molecule, and that's going to not pass the cell membrane. So not capable of passing the cell membrane. And you're going to see when we talk about lipid derivative or steroid derivative hormones, those are going to be the lipid-soluble substances that can freely pass. So steroid hormones are called lipid derivative hormones, and they freely pass the cell membrane. So the reason I'm bringing this up, we're going to get to it later, but the locations of receptors don't have to be on the plasma membrane. They can be on different membranes as well. They can be on membranes inside the cell, right? Like the rough ER, the smooth ER, those are made up of membranes. The Golgi apparatus, that's a membrane. That's the location where receptors can be as well. And so lipid derivative or steroid hormones, those are going to bind receptors that are inside the cell, where peptide hormones are going to bind to receptors that are on the plasma membrane. So they technically bind to the outside of the cell because they're binding to a membrane protein. All right, but we're going to get to that concept later. I just want to talk about it briefly, so when we get there, you're like, oh yeah, he's mentioned this quite a few times. I try to do that. You're going to see that in my lectures. I try to organize the lectures like in a logical order, covering simpler concepts and then expanding into the deeper and more complex. And then by the end, hopefully I've introduced these minor concepts along the way that once we get to the end, you're like, oh, I've heard this already and it makes sense. So we'll see this heavily at the end. All right, so we're going to expand a little bit on amino acid derivatives. Again, these are really small hormones, right? So they're going to be small hormones because they're derived from a single amino acid, right? And so small molecules, or if you want, you can just say small hormones that are derived from an amino acid, all right? There's two types of amino acids, and those two types, I think I got the question mark and the period reversed here. But the two types of amino acids you should be aware of. We have amino acid number 1, tyrosine derivatives. Now, you need to be able to identify these two categories very specifically. So categorization in this chapter is very, very key. It's the thing you need to get down. You need to spend time figuring out these. And I'm going to point out some of them. All right, so the first one, the first amino acid tyrosine derivative hormone to know is thyroid hormone. This is a must know. We're going to talk a lot about thyroid hormone. It's going to be one of the main glands that we spend a lot of time breaking down. We're going to get into its histology, so get familiar with this one. Star this, make sure you know it's really important. Now notice, this I'm telling you is a lipid-soluble hormone. Now over here, all lipid derivatives are lipid-soluble. All peptide hormones are water-soluble. They're not going to pass the membrane. The amino acid derivatives, the thyroid hormone, this is the only lipid-soluble amino acid derivative. All the other amino acid derivatives we talk about are going to be water-soluble. All right, but for now, no, thyroid hormone regulates metabolism in the cells, i.e. helps with energy, being able to create that ATP. All right, that's what you need to know for now. We're going to expand heavily on this. All right, we also have a second class of tyrosine amino acid derivative hormones, the tyrosine derivative catecholamines. Now notice these are water soluble. The main one to know is norepinephrine and epinephrine. So no nor hyphen, it's norepinephrine and epinephrine. That's both of them. This is the fight and flight response. So when we get when you get frightened, or when we activate that and your heart starts racing, that's going to be due to the release of norepinephrine and epinephrine. Those are the adrenaline hormones. But notice these are water soluble, so these are going to bind to the outer membrane receptors, where this will bind to an internal membrane receptor. But we'll again get into that later. The other catecholamine to note is something called dopamine, and it's called catecholamines because they're water soluble. Dopamine, you should know, is your reward center response, right? It's often the neurotransmitter, but we need to know that there's a lot of functions overall as a hormone. You just need to be able to classify dopamine as catecholamine. That's it. We're not going to get into the function of dopamine, just be able to classify it as a catecholamine and know that it's a tyrosine derivative. All right, norepinephrine and epinephrine in thyroid hormones, those are the must-know ones. Dopamine, you just need to be able to categorize as a catecholamine. All right, the other amino acid is the tryptophan derivative amino acids hormones. And so the tryptophan derivative hormones, these are going to be, if you remember, what's the hormone? Well, where do we get tryptophan in? We get it a lot during Thanksgiving because of turkey, right? Well, tryptophan, that increases your, what's the hormone that makes you sleep? Well, melatonin. So tryptophan and melatonin, you should be able to associate it with melatonin. That, of course, is the sleep hormone that regulates the day cycle. You should also associate the tryptophan derivative as serotonin, and note that that's a mood enhancer. It has a lot of other functions, kind of like dopamine being the reward response, serotonin being the mood enhancer. There's a lot more functions behind them. They're some of the main neurotransmitters of the brain, but understand that they also can function as hormones as well. We don't spend a lot of time with them. At least I don't. So if you're in a different course, make sure you know what your instructor wants you to know about dopamine and serotonin. For my course, I don't expect my students to know too much here. All right, but overall, because amino acid derivatives are small molecules, literally an amino acid with some other chemicals attached to them, understand these are the short-lived molecules. They're going to survive for minutes to hours, and they are one of the shorter-lived versions of any of the hormones. These do not last long because they're small molecules. The smaller the molecule is, the more likely it breaks down very quickly. All right, peptide, this is our second class of hormone. These are going to be longer-lived hormones compared to the amino acid derivatives, but they still are going to be short-lived, so minutes, hours, and even days in our bloodstream. Again, keep in mind the endocrine system maintains hormone concentrations, and those hormones, they're basically going to bind to receptors when the receptors become available and keep our responses normal. This will start to make more sense as we get deeper into the chapter and start talking about the various types of hormones. All right, so lipid soluble or water soluble, if you saw, probably would have been able to see that. Of course, these are peptides. Again, peptides are large molecules, and they're made up of many amino acids. Amino acids have polar charges to them, so in this case, you should know these are water soluble. All right, and simply what that means is these cannot freely pass the membrane. They're going to have to bind to what's called an extracellular receptor. And we're going to define extracellular receptors, but these are receptors that are on the plasma membrane and sticking in the extracellular fluid, which means it's on the outside of the cell. So peptide hormones always bind to extracellular receptors. This is a huge concept that we're going to build off of, but we'll get to that more specifically by the end of the chapter. All right, oftentimes peptide hormones are produced as prohormones. Anytime you see a prefix pro in front of a chemical that you might be familiar with, what that simply means is it's going to be an inactive molecule that becomes active. And so an example is when insulin is produced, it's produced as proinsulin. That allows the cell to have pretty much insulin ready to go. It just needs some minor modification. All right, and that's important because, of course, like after we eat a big meal, all of a sudden our blood glucose is going to shoot up. We don't want to have to go through the whole process of transcription, translation, and then producing the protein and then modifying the protein. And these prohormones are already produced. They just need one minor signal to say, all right, we need the actual insulin ready to go. And in that case, they would just modify the actual protein already, and insulin's free. It's not a big deal that you know that. That's what's going on here, but just understand prohormones are inactive versions. We'll see that. We'll develop off that a little bit later. Now, with peptide hormones, all peptide hormones are going to be the primary messenger. In fact, any water-soluble hormone is the primary messenger, and what that means is it's the first molecule that's going to start a chemical cascade that's going to result in a response. Now, if you're starting to get a little confused here, that's okay. It is a tough introduction here. We're sort of getting into some of the tougher parts that I'm going to build off of later. It's got a lot of key terms that you're going to have to work through, but a primary messenger starts off something called a chemical cascade, and what that is, it's a series of chemical changes that's going to eventually result in a cellular response. Our bodies are really complex because we create lots of enzymes. The more enzymes and more chemicals that have to be created for a process to complete, the more control our body has over things. So, maybe it's 8 chemicals that have to be changed, and then eventually that 8th chemical actually carries out the response. That's what a chemical cascade is. It'd be chemical 1 becomes chemical 2, chemical 2 becomes chemical 3, chemical 3 to 4, all the way down to 8. When we reach chemical 8, a cellular response occurs. Well, peptide hormones are the first chemical that start this chain reaction, so they are called the primary messenger. They start the chemical cascade. Okay, so for now, just know these terms. Start thinking about what a chemical cascade is. We're going to expand on this a lot, but basically, these are the major points of peptide hormones to really understand. They're longer lived in comparison to the amino acid derivatives because they're composed of many amino acids. You need to know these are made up of amino acids. Know that they're water soluble. Know that they bind to the extracellular receptors. Note that they start the chemical cascade. Okay, so work through these definitions. It's a lot to ask, but these are important ones. All right, so that's the second main class. And then we get to lipid derivative chemicals. Now, these are lipid derivative chemicals, but really what we're going to do is we're going to focus on the two types of lipid messengers, which are lipid soluble. Again, what lipid soluble means before we get into the two types. Lipid soluble means they can pass the cell membrane, so these chemicals will pass the cell membrane, and they're going to bind to receptors inside the cell. We were using the term extracellular receptors for peptide hormones. These would be intracellular receptors, so inside the cell, intracellular. Again, concepts that we'll build off later. I just want to introduce them so you start getting familiar with those terms. Now, the main lipid derivative messenger we're going to deal with is the steroid hormones, aka steroids. You should note that these are derived from cholesterol. It's really important. This is why maintenance of cholesterol levels is important. It's also important for your cell membranes, but also this is why it's important to take non-steroid anti-inflammatory drugs, NSAIDs, over. This is why something like prednisone, which is a really strong, it's a really strong anti-inflammatory. But the reason you have to wean off of it is because it's a steroid medicine, and so that steroid, this being able to kind of share a chemical structure, makes things a little bit more complex. All right, and because they're a steroid, you should note they are longer lived. These can last days to week, and guess what? Many of these hormones are going to be the ones related to puberty, right? Because puberty, as I said, is going to be a multiple year-long process, and so these are going to be long-lived hormones. All right, and then the final thing to note that we will again expand on later, these are going to be called bound hormones. When anytime a steroid or a lipid-soluble molecule, if it's lipid-soluble, it has to be bound to a protein when circulating, because proteins, again, cannot just readily pass the cell membrane. And so by being bound to a protein, that's how lipid molecules can circulate, because otherwise they're going to go pass through cell membrane after cell membrane after cell membrane, which means it would be impossible for them to reach their destination. So often they're going to need circulating proteins, which we'll talk about later. So some of the main points, definitely know derived from cholesterol, and know that these are the longer lived ones. And then off to the side, you might just want to put hormones associated with puberty, all right, or they're called sex hormones. You're going to want to put those because the way you're going to want to learn which hormones are which, you're going to want to learn the steroid hormones and then just assume everything else is a peptide hormone except for thyroid. Okay, that's kind of the approach you take. Now, of course, that means you still need to figure out the amino acid derivatives a little bit more strongly, but most hormones are going to be peptide based. I think there's 7 steroid hormones. Now, the other type of lipid derivative chemical messenger is the eicosanoids, and if you remember, those were paracrine signaling. So we're building off of something that was introduced early on in this class, right? It was one of the first slides. So you should be able to associate eicosanoids with paracrine signals, and these are the 2 eicosanoids I expect you to note and expect you to know what they're doing. If you remember, I said it was often associated with repair processes, or in this case, blood clotting. So notice prostaglandins, blood clotting via vasoconstrictors, and then notice the leukotrienes. These are the eicosanoids that stimulate inflammation. This is the extent of what you need to know for eicosanoids. Know that they're paracrine signals, they're derived from something called arachidonic acid, and be able to recognize that these 2 chemicals are involved in the blood clotting process. All right, so don't get too caught up on eicosanoids. We're going to be focusing most of our attention on the steroid hormones, but I expect you to know minimally when you see the word eicosanoid, you should be able to recognize that it's a paracrine signal. In fact, on the exam, I'm heavily considering doing various types of matching, and as far as those signaling goes, autocrine, endocrine, synaptic communication, I can easily match those. And when it comes to being a paracrine signal, I can use either prostaglandins, leukotrienes, or the word eicosanoid, and all of those you would need to know relate to our paracrine signals. Okay, so those are the sort of things when you're listening to content. Think about how I can organize them, and I just gave away a good example. Eicosanoid, prostaglandin, and leukotriene in a matching sort of question to make sure you know that that's a type of paracrine signal. All right, so steroid hormones, just to expand on these just a little bit, we're kind of wrapping up here. We are, like I said, this was one of the longer lectures because I had a lot to introduce you to. Hopefully you're starting to see the layout and the color code. So again, I'm about to define steroid hormone. All right, but steroid hormones, these are lipid messengers derived from cholesterol. All right. And again, they circulate as bound hormones. Remember, we need to bind a protein to them because they're a lipid-soluble molecule. And what that means is they are attached to a transport protein. All right. No steroid hormones. These are a lot harder to break down, which means they last longer in circulation. So if I ask any question about which of the following hormones breaks down the fastest, you would know it's going to be your amino acid derivatives. Otherwise, which of the following hormones stays in circulation the longest? It's going to be a steroid hormone. So know your steroid hormones. I can even put up as an answer choice steroid hormone, or I can just list the steroid hormones themselves. All right, but these are harder to break down when they're bound. Remember now, enzymes must break down, in this case, if they're bound to something, enzymes only function on one molecule. So until it's unbound, enzymes can't even break these components down. All right, main lipid-soluble hormones so far. All right, so I'm just to kind of reiterate what are the lipid-soluble hormones we've discussed. You should know thyroid hormone is a lipid-soluble hormone, and we have our steroid hormones. And so our steroid hormones, just to give you the list right now, and I would start working on getting this to memory, but your major list of your steroid hormones are going to be the reproductive organs, so any of the sex hormones we talk about, and then hormones that come from the adrenal cortex. That's going to be aldosterone, cortisone, and androgens. And then one hormone from the kidney known as calcitriol. Now the sex hormones, if you didn't know what they are, it's testosterone, progesterone, and estrogen. So you got 3 sex hormones to know, you have 3 adrenal cortex hormones to know, and you have the calcitriol from the kidney to know. If you get those 7 to memory, the remaining hormones are going to be water soluble, except for thyroid, because thyroid, if you remember, is an amino acid derivative. Thyroid is an amino acid. It's a tyrosine derivative, which is what you should know for it. All right, but again, lipid soluble, what defines something as lipid soluble? Non-polar molecules that can freely cross the membrane when not bound to a protein. So again, lipid soluble molecules will be transported bound to a protein is your key point, key walkway to make sure you understand. It's a concept that we're going to continue to build off of. All right, so we skipped over a lot of figures. There really wasn't a lot of figures to start off, but I think Pearson does an amazing job with all of their figures. I encourage students, as you feel confident with the content, you should look at a figure right here. Where do your eyes catch? Oh, amino acid derivative. Can you just, if you saw that, can you identify tyrosine? And if you did, could you, instead of seeing thyroid hormones, could you think thyroid hormone and catecholamines? All right, can you think melatonin for tryptophan? Okay. Notice peptide hormones, they expanded on them, talked about various glycoprotein. I don't care about these. You don't need to expand to know glycoprotein versus short polypeptides. We're just going to stick with peptide hormones. I'm not doing these minor subclassifications here. You can see your lipid derivative. You have your steroid hormones being the main thing we focus on. It's this shape. And then notice your eicosanoids. They're just telling you local paracrine factors. So that's what you should associate it with. All right, the final concept to close out of this, because we just were talking about this, I think it's important that I really close out on this concept. It was a big one, which is water soluble versus lipid soluble. And so water soluble are polar hormones that cannot pass the bilayer, and therefore they bind extracellular receptors. What are extracellular receptors? Well, if you remember, I was saying receptors that bind to hormones on the outside of the cell. These are hormones that can't cross the cell membrane, therefore it's like putting something in your mailbox and then the mail carrier taking it onward. Well, the mailbox is kind of that receptor. All right, all peptide hormones are going to be binding to extracellular receptors, so all peptide hormones are going to be primary messengers, right? Because again, these are primary messengers. All amino acid derivatives except for thyroid hormone. So that's how you break this down. These are going to be the water soluble. And then these are going to be free hormones. They're hormones that are never bound to transport proteins, which makes them easier to break down because, again, they're a single molecule, which means enzymes will exist to break them down independently. And really quickly, sorry, just to clarify something, I was mentioning that these are primary messengers. All of these are primary messengers. Any hormone that binds to an extracellular receptor is a primary messenger. That's what makes it a primary messenger, is it binds to extracellular receptors that causes a chemical cascade. All right, so overall, and this is the big picture stuff you need to really understand about water-soluble hormones. If you can get these details down, which you need to review this multiple times to really get it down, but you can come back and revisit this as we move along and get deeper into the chapters. All right, so what makes a hormone lipid-soluble? So when we talk lipid-soluble chemicals, these are going to be nonpolar chemicals. In this case, we're talking hormones, so nonpolar hormones that can freely pass the bilayer. And because they pass the bilayer, that means they're going to bind to hormones on the inside of the cell, which are known as intracellular receptors. All right, so intracellular receptors or receptors inside the cells. All right, these are going to be all steroid derivative hormones and thyroid hormones. So that's what's going to be lipid soluble. All steroid derivative hormones are lipid soluble and thyroid hormone. And all of these will also be considered bound hormones. So where these are free because they're polar, they're not crossing cell membranes freely, so they can just travel in the bloodstream because they're not going to be passing and essentially leaving the capillaries because they're polar, so they can't just pass the epithelial cells. Where these, because they're nonpolar, they need to be bound to a protein because literally they would be leaving and passing right through the capillary and just moving on. So that's why they need to be bound. It keeps them within the bloodstream. All right, so students need to get in the habit of categorizing each hormones and knowing water or lipid soluble. So not only do you need to know are they considered steroid derivative, peptide derivative, or amino acid derivative, you also need to know water soluble and lipid soluble. Like I said, the best approach is learn your learn your steroid hormones, because there's only 7 of them, 3 of which come from sex organs, 3 of which come from the adrenal cortex, and only 1 comes from the kidney. Those should not be hard to get through, but this is going to take practice. This chapter overall, I think, is a rather straightforward, direct chapter. It's just a lot of content that you need to sift through. So do not delay studying this. Get familiar with these terms. If you know there were terms that weren't making sense, start writing those terms off to the side to say, hey, I need to work this term out. Because if a lot of these, what makes this chapter very hard is we have terms like intracellular, extracellular. We have the words bound versus free. A lot of this is perspective on what I'm talking about. And I can use a lot of these terms interchangeably, which often makes it hard for students to follow because you really need to grasp how there's just plenty of ways. Like I said, lipid soluble and steroid derivative have a very similar meaning to me. Now, water soluble doesn't have the same kind of meaning in comparison to something like peptide hormone, because water soluble or because lipid soluble and steroid derivative are so tightly linked, where water soluble, a lot of chemicals are water soluble. Anyway, don't get too caught up on what I just said there. It's not really that relevant here. All right, so if you notice at the end of my lectures, I will close out with the basic group note reviews. What these are is I feel like if students can address the vast majority of this, that means they grasped the lecture. So like if you didn't have to use your notes and you can kind of define all of these and explain them, you probably really understand it. If this seems foreign or you're like, I have no idea where this content came from, that means you missed a lot of the big picture content throughout this lecture. Overall, you should know these top 3 right now because that's where we're at. These bottom 4 is what's going to be covered later. So if you feel comfortable with these top 3, I would say it's pretty good to move on to the next part of lecture 1. If you're fairly confused by this, take note and say, I'm probably going to have to visit some of these concepts. And make sure you do it sooner than later. You should get in a habit of right before like going to bed or calling it a night, just reviewing your notes. And I'm not saying study, I'm saying just glance over your notes and say, hey, do I remember some of these concepts? There's research that shows that even just that quick glance will help retain a lot of content. Also, as a reminder for my students, do not forget to check out those study videos that I have. I have a whole section in my module, the pre-modules. If you go to the study help start here sort of thing, you should see that I have some really good information about study tips and study habits. So I would encourage you to read or to review that little mini series. And I'll remind you probably in not in lecture 2, but probably sometime in lecture 3 about this. But I am going on 50 minutes. This is a lot longer than I wanted to go on, but usually some of these intros have to be long, especially because I had to talk about the color coding and how the figures and how everything's laid out, which I'll continue to remind you on the next few parts. But soon lectures should become consistent, and you kind of just get familiar with my style. I'm very consistent with this kind of layout. All right, but thank you all for listening. I know this was a long one. I hope the next one will be a little bit shorter, but endocrine system is kind of a beast. This first unit is really a beast of a unit because it's more of the more of how cells function and work than the anatomy connected to physiology, which we will get into when we really get back to the cardiovascular. So bear with me on Unit 1. It's a lot of physiology here. But that is the difference between A&P 1 versus the second semester of anatomy, which is A&P 2. Take care, everyone, and I look forward to seeing you at the next part of this lecture. We will close out Lecture 1 with Part 2, and I think it's a little bit easier to digest, but Part 1 certainly had a lot of key terms. See you at Part 2. Take care, everyone.